All right, welcome. Today we are diving deep into the adaptive immune response, and we're going to focus on the story of one of its most important soldiers, the T cell. We're going to cover this in the kind of detail you'll need for a university-level immunology exam, following the entire journey of a T cell from the moment it senses danger all the way to the final command to stand down. Okay, let's get into it. So here's our game plan. First, we're going to look at something called the Major Histocompatibility Complex, or MHC. Think of it as the display case that our cells use to show off threats. Then, we'll get into the nitty-gritty of how a T cell gets activated and decides what it's going to be when it grows up. After that, we'll explore the different kinds of T helper cells, the specialists. Then we move on to the cytotoxic T cells, the actual killers. And finally, we'll wrap up with how this incredibly powerful system is kept in check and what happens when that regulation fails. Okay, so let's start with the most basic problem the immune system has to solve. T cells need to see what they're supposed to be fighting. But, they can't peek inside your other cells to see if a virus is hiding in there. They need other cells to act as scouts, to process evidence of an invader and then present it. This whole system of displaying evidence is called antigen presentation. And the molecular billboards that do the presenting are called the Major Histocompatibility Complex, or MHC. And this, right here, is the central question. This is what the whole MHC system is built to answer. Think about it. If a virus is replicating inside one of your cells, that's an intracellular threat. Sending antibodies after it, totally useless. They can't get inside. The only real solution is to kill that infected cell. But what if the threat is a bacterium just floating around in your blood? That's an extracellular threat. Killing your own cells won't help. You need a totally different strategy, like making antibodies to mop up the bacteria. So the type of MHC presentation is the first crucial decision point that tells the immune system which playbook to use. To handle this, the body has evolved two separate but brilliant display systems. For threats on the inside, like a virus that's hijacked a cell's machinery, we use MHC class I. This is basically the infected cell raising a flag that tells a specific kind of T cell, a CD8+ cytotoxic T cell, hey, the problem is in here, you need to take me out. Now for threats on the outside, things like bacteria that have been gobbled up by specialized immune cells, those cells use MHC class II. This presents the evidence to a different T cell, a CD4+ helper T cell. It's like a scout showing its commander. Look what I found out there, we need to organize a bigger response. All right, this table is pure gold for any exam, so let's walk through it piece by piece. First up, presence. MHC class I is found on all of your cells that have a nucleus. Why? Because literally any of them could get infected by a virus. MHC class II, on the other hand, is only on professional antigen-presenting cells, or APCs. Think macrophages, dendritic cells. Their whole job is to find external threats. Next, presents II. This is a rule you just have to know. Class I presents to CD8+ T cells, the killers. Class II presents to CD4+ T cells, the helpers. Easy memory trick. 8 times 1 is 8, 4 times 2 is 8. Now, antigen source. For class I, it's endogenous, proteins made inside the cell. For class II, it's exogenous, from stuff captured outside. Peptide size. Class I's binding groove is like a hot dog bun with closed ends. It holds a small, specific peptide. Class II's groove is open on the ends, so it can hold longer, floppier peptides. Structure. Class I is an alpha chain paired with a little guy called beta-2 microglobulin. Class II is just an alpha and a beta chain. The genes in humans are called HLA genes. This genetic diversity is why finding a matching organ donor is so hard. Your T cells are trained to see your specific HLA pattern as self. And finally, the pathway, which we're about to detail. For class I, it's the proteasome pathway. For class II, it's the phagolysosome pathway. Okay, so let's trace this endogenous path. Imagine a virus has infected one of your cells. Step 1. The cell's proteasome, which is basically its protein garbage disposal, starts chopping up not only the cell's old proteins, but also the new viral proteins. Step 2. These little protein fragments, or peptides, get pumped from the main cell area into a compartment called the endoplasmic reticulum, or ER. The pump that does this is a protein called TAP. Step 3. Inside the ER, brand new MHC class I molecules are being built and are just waiting. A viral peptide gets loaded right into the MHC's binding groove. And step 4? This whole package, the MHC molecule holding the piece of the virus, is shipped to the cell surface, where it's put on display like a warning flag for any passing CD8+ T cell to see. Now for the other side of the story, the exogenous path.
An APC, like a macrophage, eats a bacterium. It gets trapped in a deadly bubble called a phygolysosome and gets digested. Meanwhile, in the ER, MHC class II molecules are being made. Now here's a super clever part. To stop these MHC2 molecules from accidentally picking up the cell's own internal peptides, which are meant for MHC1, their binding groove is plugged up by a placeholder protein called the invariant chain. This vesicle, with the plugged-up MHC2, then travels and merges with the phagolysosome that's full of chewed-up bacteria. In that acidic environment, the invariant chain is destroyed, opening up the groove. A piece of the digested bacterium is loaded in, and this final complex gets sent to the surface to show CD4+ helper T cells what's going on. Okay, so our antigen-presenting cell is now displaying a piece of a pathogen, but just seeing that isn't enough to launch a full-blown immune response. Activating a T cell is a huge deal. If you get it wrong, you could trigger an attack on your own healthy tissues. So to make sure it only happens when it's absolutely necessary, the body uses a super secure 3-signal activation system. Let's break down this 3-key system. Signal 1 is all about specificity. The T cell receptor has to be a perfect match for the specific MHC peptide complex being shown. It ensures the response is laser-focused. But here's the crucial part. If a T cell gets signal 1 and only signal 1, it doesn't activate. It actually gets shut down permanently. This is a safety feature to prevent reactions to self-antigens. Signal 2 is the confirmation signal, the danger signal. It's a second handshake, usually between the CD28 protein on the T cell and a protein called CD80 or 86 on the APC. APCs only put out these CD80/86 proteins when they've actually detected a real pathogen, so this confirms it's not a false alarm. And finally, signal 3 is the differentiation signal. This is where the APC releases chemicals called cytokines that basically give the T cell its mission orders, telling it what kind of enemy it's up against and what kind of warrior it needs to become. And this slide perfectly shows the power of signal 3. A naive CD4+ T cell is like a fresh recruit with no specialty. It's at a crossroads. If the cytokine environment is rich in interleukin-12, that T cell gets its orders to become a Th1 cell. Th1 cells are the generals of cell-mediated immunity. They're all about activating other cells to do the killing. But if that same naive T cell finds itself in a soup of interleukin-4, it gets pushed down a completely different path. It becomes a Th2 cell, a specialist in humoral immunity, the part of the immune system that fights with antibodies in your body's fluids. Same starting cell, totally different outcomes, all dictated by that third signal. So once that T cell gets its orders, how does it lock in that decision? It does it using things called master regulator transcription factors. For Th1 cells, the boss is a protein called TBET. When TBET gets turned on, it flips the switches on all the genes a Th1 cell needs to do its job. And just as importantly, it goes and actively turns off the genes for the other T cell types, especially the master regulator for Th2 cells. And the counterpart for T2 cells is GATA3. Just like TBET runs the Th1 show, GATA3 is the master of the T2 world. Now, what's really fascinating is that these two are mortal enemies. TBET actively blocks GATA3, and GATA3 actively blocks TBET. This creates this really strong polarizing effect. Once a cell commits to becoming a Th1, TBET makes sure it can't ever second-guess itself and try to become a T2. This ensures the immune system commits fully to a single clear strategy instead of a confused weak one. Okay, so our naive T cells have been activated, they have their marching orders, and they become specialized effector cells. They're no longer rookies. Let's meet the special forces, the major players in this T helper arsenal. First up, the Th1 cells, the generals of cell-mediated immunity. Their main weapon is a cytokine called interferon gamma. This stuff is like a super soldier serum for macrophages. It turbocharges them, making them way better at killing pathogens they've eaten. Th1 cells also produce IL-2, which is like a high-energy fuel that helps our army of CD8+ killer T cells grow and multiply. Next are the Th2 cells, the directors of the humoral response. They release IL-4, which is the key signal to tell B cells to start pumping out antibodies. It also encourages B cells to switch to making a specific type of antibody, IgE, which is really important for fighting off big parasites like worms and is also the main culprit in allergic reactions. And another one of their cytokines, IL-5, is the go signal for another cell type, the eosinophil, which is a parasite-killing specialist. And we can't forget the Th17 cells. Think of these as the first responders who call in the cavalry. Their signature cytokine, IL-17, is an incredibly powerful signal that screams, "We need neutrophils over here right now." And neutrophils are the foot soldiers of the immune system, great at clearing out bacteria and fungi. But there's a dark side. All that inflammation can cause a lot of collateral damage. In autoimmune diseases like rheumatoid arthritis, it's rogue Th17 cells that keep calling neutrophils into the joints, causing the chronic damage and pain.
So we've talked a lot about the CD4 plus helpers, the coordinators, the generals. Now let's turn our attention to the CD8 plus killers. These are the agents who carry out the executions, responsible for directly eliminating any of your own cells that have been compromised by a virus or have turned cancerous. A fully activated CD8 plus T cell is now called a cytotoxic T lymphocyte, or CTL. Its job is to patrol your body, checking the MHC class 1 molecules on every cell it meets. And if it finds one displaying a foreign antigen it recognizes, that kill me signal we talked about, it carries out a very precise, targeted kill. It doesn't just blow things up. It induces something called apoptosis, or programmed cell death, which is a clean, quiet way for a cell to self-destruct, minimizing damage to its healthy neighbors. So, how does it do it? The CTL has two main weapons. The first is the perforin granzyme pathway. The CTL latches onto its target and releases a payload from its granules. The first protein, perforin, acts like a molecular hole punch, poking holes in the target cell's outer membrane. This allows the second protein, granzyme, to sneak inside. Once in, granzyme flips the switch on a series of enzymes called caspases, which kicks off the cell's self-destruct program. The second method is what you can think of as a deadly handshake. The CTL has a protein on its surface called Fas ligand. Many of our cells, especially if they're stressed or infected, will put out the matching receptor called Fas. When the Fas ligand on the killer cell binds to the Fas receptor on the target cell, it's a direct signal that tells the target cell it's time to die. This binding immediately triggers that same caspase self-destruct cascade from the inside.
So we have this incredible system that can hunt down and kill our own cells. It's unbelievably powerful, but that also makes it unbelievably dangerous. Without some serious checks and balances, this system could easily turn on us and cause devastating autoimmune disease. So this final chapter is all about how we keep that power under control and the chaos that ensues when those controls fail.
The main peacekeepers of the immune system are a special kind of T cell called regulatory T cells, or Tregs. Their whole job is to pump the brakes and suppress immune responses. They have a couple of clever ways of doing this. First, they release inhibitory cytokines like IL-10 and GFB-theta that tell other immune cells to calm down. Second, and this is really neat, Tregs have a massive number of receptors for IL-2 on their surface. Remember, IL-2 is the fuel that effector T cells need to survive and multiply. The Tregs act like sponges, soaking up all the available IL-2 in the area, essentially starving the other T cells and causing the immune response to fizzle out. But what happens when a pathogen figures out how to hack the system? That's what a superantigen does. A normal antigen is very specific. It might be recognized by, say, 0.01% of your T cells. But a superantigen is different. It doesn't fit neatly into the MHC groove. Instead, it acts like a piece of molecular glue, binding to the outside of the MHC and T cell receptor and forcing them together, regardless of what they're specific for. This bypasses all the normal safety checks and can cause a massive, system-wide activation of 20% or more of all T cells in the body. As you can see from this chart, that's a catastrophic difference.
And this massive activation sets off a deadly domino effect. With so many T cells activated at once, they release an overwhelming flood of pro-inflammatory cytokines, creating what's known as a cytokine storm. This storm triggers systemic inflammatory response syndrome, or SARS, where your entire body's inflammation systems fire at once, causing high fever and leaky blood vessels. This can quickly progress to toxic shock syndrome, with a catastrophic drop in blood pressure, and ultimately multiple organ dysfunction syndrome, as your vital organs start to fail. It's a terrifying showcase of the immune system's power turned back on itself, and it brings our entire story full circle, from antigen presentation and T cell activation right through to the devastating system-wide consequences.